Germination. Introduction. Germination in plants is the process by which a dormant seed begins to sprout and grow into a seedling under the right growing conditions. Absorption of water, the passage of time, chilling, warming, oxygen availability, and light exposure may all operate in initiating the process. In the process of seed germination, water is absorbed by the embryo, which results in the rehydration, softening, and expansion of the cells. Shortly after the beginning of water uptake, that is imbibition, the rate of respiration increases and various suspended or much reduced dormant stage metabolic processes resume. These events are associated with structural changes in the organelles in the cells of the embryo. Germination sometimes occurs early in the development process. This process influences both crop yield and quality. It starts the growth process by activation of enzymes. The seed starts to also produce proteins and metabolizes the stored food. By rupturing of the seed coat, radicle emerges to form a primary root. The seed starts absorbing underground water and forms plumule. After the emerging of radicle and plumule, shoot starts growing upwards. In the final stage of seed germination, the cells of the seeds become metabolically active, elongates and divides to give rise to the seedling. Types of germination: epigeal germination, hypogeal germination. Special type of germination: viviparous germination. Epigeal germination. In this, the cotyledons are raised out of the soil and generally become green and photosynthetic. In dicots, they are pushed up by rapid extension of hypocotyl before growth of the epicotyl. Epigeal germination occurs in bean, castor, mustard, tamarind, sunflower, etc. Hypogeal germination. In this type of germination, the cotyledons remain underground. Hypocotyl growth is restricted. The epicotyl grows to raise the first leaves out of the soil. Hypogeal germination occurs in dicotyledon seeds of pea, mango, groundnut, etc. and in monocotyledons like rice, maize, wheat, etc. Viviparous germination. This is a special type of germination occurring in mangrove plants. These plants generally grow in salty lakes, sea coasts, and deltas. The seed germinates while still attached to the parent plant. The embryo emerges out of the fruit with a massive radicle pointing downwards. Due to increased weight, the seedling separates from the parent plant and establishes itself in the muddy soil below. Example: Rhizophora. Process behind germination: rehydration, imbibition of water, swelling of seed. RNA and protein synthesis is stimulated, increased metabolism, increased respiration, hydrolysis, digestion of food reserves by enzymes, changes in cell ultrastructure, induction of cell division and cell growth. Pre-germination. Rupture of seed coat takes place. Emergence of seedling, usually radicle first. Post-germination. There is controlled growth of root and shoot axis and controlled transport of materials from food stores to growing axis. Senescence, aging of food storage tissue also takes place. Factors affecting germination. Abiotic factors are light, temperature, aeration, oxygen, soil type, and depth of sowing. Biotic factors are viability of seed, dormancy period. Factors that affect seed germination are divided into two categories. Internal factors. Internal factors include seed vitality, genotype, seed maturation, seed dormancy. External factors. External factors includes water, temperature, oxygen, light, smoke. Light. Generally, seeds require darkness to germinate. However, lettuce, tobacco, tomato, and many grasses need light exposure to germinate. These seeds require the red portion of the light spectrum, while the far red light inhibits germination. Many small seeds with low amounts of storage reserves, such as lettuce, show such a red light requirement. These seeds must not be buried below the soil so deeply that the light cannot penetrate. Phytochrome is a plant pigment found in the cytoplasm that senses the presence of red light. The red absorbing form of phytochrome changes to the far red absorbing form when it absorbs red light of 600 nanometer and back again when it absorbs far red light of 730 nanometer. Temperature. As with most reactions, germination generally occurs faster when at warmer temperatures. Sometimes there is a need for cool temperatures to break dormancy. Stratification is one strategy that is employed in woody species in particular. It requires a moist, cool period that degrades growth inhibitors that prevent germination. Once the inhibitors are degraded and all other conditions are met, then germination will occur. The temperature for germination to occur is quite different than the temperature requirement to break dormancy. Generally, the temperature for germination correlates to the temperature needed for successful plant growth. Seeds of cool season crops germinate best at temperatures between 32 and 50 degrees Fahrenheit. Examples: celery, lettuce, peas. While warm season crops germinate best at temperatures between 59 to 79 degrees Fahrenheit. Oxygen. Another requirement for germination is aeration, oxygen. Respiration rate for germinating seeds are very high, therefore adequate oxygen is necessary. The germination percent of most seeds will be retarded if the oxygen percent goes below 20%. Normal air is 20% oxygen. Seed beds that are overwatered or poorly drained will cause the oxygen supply to become limited, so the germination percent will diminish. Water. It is extremely necessary for the germination of seeds. Some seeds are extremely dry and need to take a considerable amount of water relative to the dry weight of the seed. Water plays an important role in germinating. It helps by providing necessary hydration for the vital activities of protoplasm, provides dissolved oxygen for the growing embryo, softens the seed coats, and increases the seed permeability. It also helps in rupturing of seed and also converts the insoluble food into soluble form for its translocation to the embryo. Soil type. There are so many parameters of soil that affect seed germination. In that factors, soil salinity, acidity, soil concentrate, electrical conductivity of soil, soil porosity, water holding capacity, texture of soil also affect the seed germination. Most problematic factor is water holding capacity of soil. Depth of sowing. Proper planting depth is a direct correlation to seed size. The general rule is larger seeds can be planted more deeply than small seeds. This is due to the energy needed to emerge. Larger seeds have greater food reserves from which it can draw energy for respiration and growth. They are able to emerge from greater depths. Soil types also affect the planting depths. The surface of sandy soil tends to dry out quickly, so seeds planted in these soils should be planted deeper than in loam soils. Seed dormancy. Most seeds produced by mature plants pass through a period of inactivity or dormancy prior to germination. During this period of inactivity, seeds remain viable. Dormancy may be internal, external, or a combination of both.